Welcome back, everyone. So we just introduced the kind of different roles of our cell membranes or of membranes in general. We discussed that historical development of different research findings that led to the fluid mosaic model. And now we're really going to zoom in on what the lipids are doing, that fluid part of the membrane. So even though they are phospholipids, there is still a lot of diversity and a lot of fluidity in how the lipids are going to be in the cell. The main types of lipids we're going to see are going to be phospholipids, glycolipids, and sterols. Phospholipids are the most abundant part of cell membranes. You're probably very familiar with them at this point. They have this phosphate head that is hydrophilic, these lipid tails that are hydrophobic. So they have these two fatty acid tails, and their phosphate head is negatively charged, which allows it to be able to interact with that polar water molecule. And a charged head group attached to the phosphate. And they are amphipathic. Amphi means both. So they are both hydrophilic and hydrophobic. They are hydrophilic because of that hydrophilic head. They're hydrophobic because of those tails. That is a very important key function of our membranes, that we have a hydrophilic area that can interact with the water outside of it. But by having that hydrophobic tail, you have that barrier so cells aren't dissolving into the water outside or dissolving into the water inside because of that hydrophobic core of the membrane. The backbone can come from glycerol, which is a 3-carbon sugar, which then one of the carbons goes up to the phosphate, and then 2 of the carbons come down to your fatty acid tails. Or it could be based on sphingosine. Sphingosine is going to have part of it come down as a tail, attached to another fatty acid tail, and then attached to a phosphate and coaling there. The glycerol-based molecules of phospholipids could include phosphoglycerides, and it could include sphingosine-based phosphosphingolipids. But these are all going to be the similar kind of shape to our phospholipids, right? With our head, a charged group, and a couple of fatty hydrophobic tails. Because the phospholipids are amphipathic, although it does have a good barrier against water and it won't dissolve in water, other things that are amphipathic can disrupt membranes. So a detergent can disrupt a cell membrane, which is convenient if you're wanting to disrupt a membrane to clean something. You can clean it with a detergent. It also means in the lab, if we want to disrupt a membrane and, pull out a protein or purify a protein from that membrane, we can use a detergent to pull out or remove a protein from that cell membrane. This also gives you different kinds of phospholipids that are in different kinds of membrane. Here is what is in your plasma membrane of a rat liver. We have mainly phosphatidylcholine. We also have phosphatidylethanolamine. We have some phosphatidylserine. We have some sphingomyelin. The plasma membrane of a potato, you can see different species have different characterizations of their different types of membranes. Here's an E. coli bacterial membrane. It's got more phosphatidylglycerol and diphosphatidylglycerol cardiolipin, but also still a lot of phosphatidylethanolamine. The myelin sheath of a human, part of your nervous system. The thylakoid membrane of a chloroplast from a spinach, and the inner mitochondrial membrane from a rat liver. So the take-home message is not that you need to memorize these. The take-home message is that different cells have different types of membrane, have different types of phospholipids in their membranes, and that is one way that you can characterize it. And also, because different cells are behaving a little bit differently, we have kind of different phospholipids in different cell types. Glycolipids are formed by adding a carbohydrate to a lipid. So glyco meaning sugar, lipid meaning lipid. And some are going to be glycerol based, so they will be called glycoglycerolipids. and some are sphingosine-based, so they're called glycosphingolipids. And the most common glycosphingolipids are cerebrosides and gangliosides. So you don't have to memorize this list of what is a glycoglycerolipid or a glycosphingolipid, but it is a good idea to be able to say, oh, cerebrosides, oh, gangliosides, I've heard of those. Those are parts of my cell membrane. Glycolipids, those are found in parts of my cell membrane. When we're talking about cerebrosides and gangliosides, here's a schematic of a cerebroside. So you have your sphingosine with your sugar attached to it, and your fatty acid tail gives you this kind of chemical structure with your head group and your two tails here. The ganglioside has an oligosaccharide head group with one or more negatively charged sialic acids, and cerebrosides and gangliosides are very prominent, especially in brain and nerve cells. This sugary part at the top of it is going to be a very good kind of signal to other cells, allowing that cell to be recognized and to be able to interact with other cells. Most eukaryotes also have sterols, a lot of sterols in our plasma membrane. These, you can see, don't have that same phospholipid shape. They don't have the phosphate head. They don't have the fatty acid tails. But they are still part of our cell membrane, and they are super important. In animal cells, the sterol that we have is cholesterol. Probably heard of cholesterol. It will pack into your cell membrane, and it helps stabilize it, and it helps maintain it. So it does things like keep it fluid at the temperatures where it is going to be. It does things like keep it from bursting open if the cell is in a environment where water is trying to rush into the cell. That cholesterol helps keep that cell membrane a little bit more secure. Instead of cholesterol in a plant membrane, you're going to find phytosterols, but still that's a type of sterol. And in fungal cell membranes, you're going to find ergosterol, which is also a sterol, similar to cholesterol, but a little bit different. So all of us are eukaryotes. Cholesterol if you're an animal, phytosterol if you're a plant, and ergosterol if you are a fungus. Those are the sterols that are helping maintain the integrity and the structure of our cell membranes. There are fatty acids in almost all membrane lipids, except for your sterols, which don't have it. Your other membrane lipids are usually going to have fatty acids. They have long fatty acid hydrocarbon tails. Here's some of our different illustrations of some different types of tails you might have: palmitate, stearate, oleate, and linoleate. They can range in size from 12 to 20 carbons long. That's the general range. And that is a very optimal length to form a bilayer. And that length of these tails is usually what dictates how thick the membrane is going to be. It's usually 6 to 8 nanometers, which correlates to having 12 to 20 carbons on either side of the membrane. These long hydrocarbon tails are going to be hydrophobic, so that isn't that nice barrier that prevents interaction with polar solutes or charged solutes, that they won't be able to just go in and out of the membrane between your fatty acid tails. Fatty acids can vary a lot, though. They can have multiple double bonds. They could have single double bonds. They could have one or more double bonds. The number of double bonds can vary, or even having double bonds can be a way that fatty acids vary from each other. Palmitate and stearate are common types of saturated fatty acids. You can see all of these carbons have 2 hydrogens attached to them. So it has as many hydrogens on there as it can. All of the bonds between the carbons are single bonds. They're drawn with one line. That's different from things like oleate and linoleate, which have a double bond. See, this has a double bond here. This is a double bond, which means this carbon is only going to be bound to one hydrogen now. This carbon is only going to be bonded to one hydrogen. Because they don't have that maximum number of hydrogens, they are called unsaturated. So they do not have all the hydrogens that they started with. This one, oleate, which has one double bond, and this one, linoleate, which has 2 double bonds, it doesn't make a difference if it has more than one. They are both considered unsaturated. Having any number of double bonds makes you an unsaturated fatty acids. Omega-3 fatty acids refer to polyunsaturated carbons that have a double bond on the 3rd carbon coming out. The membrane is often asymmetrical. That means that the lipids that are present on one side of the membrane or the other side are going to be different. How saturated those fatty acids are is going to be different in the side of the membrane facing the exterior or facing the interior. Most of the glycolipids in animal cells are going to be in the outer layer of the plasma membrane. That's really important because remember we said that sugar, that glyco part, is an important part of being able to recognize and interact with other cells. So that needs to be on the outside so a cell can recognize what it has found. So glycolipids are usually found on the outer layer. It doesn't do much good to keep it inside the cell. Membrane asymmetry is usually established while the membrane is being made. And after that, it really doesn't change much. Once something is determined that is going to point out of the cell or point into the cell, that's pretty much how it's going to be. Lipids, that movement of a lipid from one monolayer to another would require its hydrophilic head going through that hydrophobic interior, which just isn't chemically usually going to happen. So transverse diffusion or flopping from the exterior side of the membrane to the interior side of the membrane is a pretty rare thing. It can occur, it does occur, but it doesn't occur a lot. So lipids are mobile within their monolayer. They can rotate, so this little phospholipid is spinning around. There can be lateral diffusion. This phospholipid has moved over here, then moved here, just kind of moves around. Both of those types of movement are random and they're fast. So it's not going to just kind of take its time. It's usually just moving around, spinning, moving where it needs to move, how much energy it has, how warm it is. It's going to determine, how quickly they're moving, how they're moving. Phospholipid flip-flop can occur naturally. This is going to be rare, that kind of transverse diffusion, moving from here to here. Although it does happen, that usually takes something called a flipase. We discussed the role of flipases in a cell undergoing apoptosis, how it will create flipases to be able to specifically put markers on the outside of those little apoptotic bodies so they can be recognized. So we do have things like this happening. In the smooth endoplasmic reticulum, there are proteins that will catalyze flip-flop of membrane proteins. There, as a smoothie R is creating the final version, helping to make sure that everything is oriented correctly before that membrane goes to the cell membrane. You can actually see in a lab experiment, you can see how fluid the lipid bilayer is. So it is allowing lipids to move. It's allowing proteins to move. They can move as much as several micrometers per second. And the way that we can actually demonstrate this is a technique called FRAP, or fluorescence recovery after photobleaching. So you start off with the cell membrane. It's unlabeled, and you're going to stick a fluorescent dye all over it. So all of those molecules on the surface of the cell are now labeled. They're going to fluoresce. Here they're labeled, they show green. Here they're looking at a specific protein in the junctions of cells, so you can see it at these cell edges. Then they shine a laser beam, which is doing the photo bleaching. It's going to bleach out a patch of those molecules, so you end up with a bleached out spot. If the cell's molecules were not moving, you would expect that bleached out area to stay. If things start moving around, they're going to start being able to fill it in, so that area will fill back in. So you're able to see, after a few seconds, we're starting to see some fluorescence come back. Things have started to move into that area. So the parts of the molecules that weren't photo bleached are moving into this bleached area, and the parts that were bleached move back out. You start to see it coming back in, giving a fluorescent signal. That indicates movement of the cell molecules. Eventually, that lateral movement is going to fill in that bleached area. And you can actually get a measurement of how much has moved. So you start by measuring, before you've bleached it, how much fluorescence is there. That's your 100% limit. Then when the bleach is applied, that is your 0% limit. And then over time, you say after 20 seconds, after 40 seconds, how much of that previous layer is it fluorescing again? And you come up with being able to see, here's the path of fluorescence coming back. This increase in fluorescence shows you how much of the plasma membrane molecules have moved. and how much are immobile. How much is moved back in as a mobile fraction, and how much it's still missing. It has yet to do with the immobile fraction. Another thing that's really important about our fluid mosaic model is it has to be fluid. If a membrane is frozen, it is not going to function. So fluidity of a membrane, just like anything, is going to be affected by temperature. As the temperature gets colder and colder, it gets less fluid as it starts to get closer to being a solid. As it gets warmer, it gets more and more fluid as it's more and more melted, warm, flowing around. And all lipid bilayers have a characteristic transition temperature, which is called Tm. Tm is the temperature where it becomes fluid. So here, as the temperature is getting warmer, this is the Tm for this cell membrane. When it's colder than that, it gets more and more solid. And when it's warmer than that, it gets more and more fluid-like. So this is the phase transition when it goes from being solid to liquid. You can even see, when they're more solid, they're packed tight against each other, kind of frozen in place. And when they're more liquid, they're just kind of hanging around, moving, dancing, a little bit more spread out. If you are below the Tm, if your cell has started to get a little bit more frozen, there's a lot of functions that it's going to lose. There's a lot of functions that are reliant on that membrane fluidity because it might require a protein moving, it might require a protein opening up and receiving a signal and being able to move. Those things are not going to happen if the membrane is not fluid. Those structures, as they become frozen, will not be functional. So our cells put a lot of effort into making sure that they stay fluid. One thing that affects how fluid a cell can be, that cell membrane can be, is the fatty acid composition. As hydrocarbons get longer and longer and longer, they're going to be more and more willing to interact with each other and have stronger interactions and be more solid. So as you have an increasing number of carbon atoms in those fatty acid tails, you have a warmer and warmer transition temperature. So these that have a shorter fatty acid tail are going to be fluid at cooler temperatures. A longer fatty acid tail means it's going to take a lot hotter for it to melt. So long chain and saturated also makes a difference. As you start having unsaturated fatty acids, they don't pack so well together. Let me come back up a few slides for a second. So see these that have these double bonds, they kind of get a little bit of a kink, a little bit of a curve in it. That means it's hard for them to pack tightly to form a tight, solid crystal. These that have no double bonds, nice and straight, they can pack really quickly. So these are going to have an easier time being solid, and these are going to have an easier time being liquid. So if we go back to that slide we were just at, as you have no double bonds, as you are fully saturated, it has a much higher Tm or much higher melting temperature. As you start introducing double bonds, it gives you a much lower melting temperature. So they're going to be much more liquid even at very cold temperatures. So if it is long chain and unsaturated, it's going to have very high Tm values. Your cell membrane is going to have to stay very hot to stay mobile. And if you start introducing shorter fatty acids and multiple double bonds, your cell is going to be much more liquid even at very cold temperatures. This kind of allows a cell to adapt to its environment. If it's warmer or colder, it can adjust the number of carbon molecules in its fatty acid tails, and it can adjust the number of double bonds in its fatty acid tails to make sure that it is properly fluid at whatever temperature that cell is growing at. So here, why saturation affects the membrane fluidity is when they are saturated, they're straight and they pack together very well. When they have double bonds, they start getting these little kinks, these little curves and bends that prevent them from packing together. So these are going to stay interacting together even when it's warmer. They're going to have a very high melting temperature. These are going to have a hard time interacting with each other at all. So they're going to have a very low melting temperature. Membranes can also vary in what kind of saturation they have. They can have a cis double bond, where the rest of the membrane is on the same side, or a trans double bond, where the rest of the membrane is on other sides, where the rest of that fatty acid tail. So a trans is going to give you something like this, which is pretty similar to that saturated shape. And a cis double bond is going to give you this, more of that kinky, curvy tail. So that means that if you have a trans fat, it is going to affect your cells pretty similar to having an unsaturated fatty acid in there. If you have a cis double bond, then it's more likely that will keep your cell more fluid, keep your cell membrane more fluid, even at those colder temperatures. Sterols also impact membrane fluidity. Their intercalation into the membrane means that it's going to be harder for those phospholipids to interact with each other. So cholesterol prevents hydrocarbon chains of the phospholipids from packing together, and so it reduces the membranes to kind of gel when they get cooler. But also it's a little bit rigid. Cholesterol is rigid by itself, so that is going to increase the TM. It's going to make it less fluid because you've got these rigid pieces. So cholesterol is doing both. It's making it have a higher melting temperature and making it have a cooler melting temperature. So cholesterol is a fluidity buffer. It allows the cells to have a little bit more variety in its range of where that cell membrane can be functional. It's a fluidity buffer. The way that cholesterol works in our animal cell membranes is pretty close to how the phytosterols or the ergosterols and other organisms function in those cells. Sterols also, as they're packing in, you can see them kind of packed in. It means there's less space in between the phospholipids. So they're going to decrease how permeable that membrane is to small polar molecules. So if a small polar molecule normally comes in between the phospholipids to diffuse in the oxygen or the carbon dioxide, and then it hit a cholesterol, it's not going to diffuse so easily. So these phospholipids, their space between them gets filled up with cholesterols, which reduces how permeable it is because it blocks that route that the ions take or the small membranes take through the membrane. Some organisms actually have something called desaturase. Desaturase is an enzyme that introduces double bonds. So if the cell has more unsaturated fatty acids and it starts to get a little bit colder, the desaturase can start adding these double bonds. It desaturates the phospholipid tails. In plants and yeast, there are temperature-related fluidity changes that are tied to the increased solubility of oxygen at lower temperatures. There's more oxygen available at the lower temperatures, and oxygen is a substrate for desaturase. So that allows these membranes to become desaturated, allowing for that fluidity of the membrane to be maintained at lower temperatures. Okay, that's it for our discussion of our lipids. We still need to talk about our proteins, so we'll come back in our next video, and we're going to start talking about what our proteins are doing in that cell membrane. I'll see you then. Bye.